Good morning, my dear students. Welcome to our zoology online class. Hope you are very happy. Dear students, today we are going to study about three important topics, three important small topics. So all these three topics are very easy to understand. So listen very carefully. And these three topics are very important for 3 mark questions and 5 mark question answers. Understand? Okay, my dear students. First, we are going to study about immunodeficiency diseases. Okay, immunodeficiency diseases. Immunodeficiency diseases means, see, you all know our immune system is made up of many organs and many components. Okay, many organs, many tissues, and many cells are present in our immune system. Okay, so here immunodeficiency disease meaning the failure of one or more component of our immune system is called immunodeficiency disease. So if any components is not working properly, is not doing their function properly, it leads to some diseases. That diseases are called immunodeficiency disease. So the reason for the immunodeficiency disease is failure in one or more components of our immune system. Understand, my dear students? Then these immunodeficiency diseases are classified into two major categories. The first category is called primary immunodeficiency disease and the second type is called secondary immunodeficiency diseases. So the immunodeficiency diseases is classified into two types. The first one is primary immunodeficiency diseases and the second one is secondary immunodeficiency diseases. And the reason for this primary immunodeficiency may be due to some genetic defects. If any problems in the genes, it leads to some immunodeficiency diseases. So that diseases are called primary immunodeficiency diseases. Understand? And the reason for the secondary immunodeficiency disease is many reasons we can say. The over radiation, infections, okay, like that many reasons are there for the secondary immunodeficiency diseases. Understand? So now we studied 2 points. First I said the definition for the immunodeficiency disease. Then I explained two types of immunodeficiency diseases and the reason for these two types of immunodeficiency diseases. Understand actually? Then, so first you are going to study about one important example for immunodeficiency disease, AIDS. Okay, so now we are going to study about this AIDS disease. Okay, the full form you all know already you have studied in your lower classes. Acquired immunodeficiency syndrome. Acquired immunodeficiency syndrome. Here, acquired immunodeficiency syndrome meaning the deficiency of immunity power. Okay, so here we are getting some immunity power after our birth. Okay, that immunity power is called acquired immunity power. The immunity we are getting during our lifetime is called acquired immunity power. So this immunity power is reduced. Okay, so the deficiency of this immunity power is leads to one disease. That disease is called AIDS, acquired immunodeficiency disease. Understand of you? See, maximum this disease is there is no treatment and there is no medicine for this disease. Okay, to cure this disease. And we all know the causative organism of this AIDS disease, HIV virus, human immunodeficiency virus. Okay, human immunovirus is the causative organism of this AIDS disease. Understand of you? Then maximum this HIV virus. Okay, in short we can say HIV virus. So this maximum this HIV virus attack and destroy the helper cells, T helper cells of our body. Here T helper cells means one of the very important cells of our immune system. Okay, so here this HIV virus attack and destroy the T helper cells. Okay, attack and destroy the T helper cell. Here the T helper cells is very important for the production of antibody. Here the T helper cells will not produce antibody, but this T helper cell playing a very important role in the formation of antibodies because this T helper cell only stimulate the B cells. When B cells are stimulated, that in B cells can produce the antibodies. So the antibodies are produced by the B cells. But to produce these antibodies, B cells should be stimulated. Okay, so to stimulate the B cells, these T helper cells are very important. Understand you? So this HIV virus infect and attack the T helper cells. So because of this reason, the T helper cells are, the number of T helper cells is reduced or decreased. Okay, so if the T helper cells are not produced properly, the B cells are not stimulated. If the B cells are not stimulated properly, it affects the antibody production. It affects the antibody production. Understand of you? Understand? So now I said one important points here. This HIV virus infect the T helper cells. So here this T helper cell only stimulate the B cell for the production of antibodies. If the number of T helper cell is reduced, the antibody production is affected. The antibody production is affected. So I hope you are understood this point. Then, see, based on the antigens of this HIV virus, the HIV is classified into two types, type 1 HIV and type 2 HIV. Type 1 HIV and type 2 HIV. Understand of you? So now we are going to study about the structure of this HIV virus. Structure of this HIV virus. Understand? See, this HIV virus belongs to a genus called lentivirus genus. Okay, the genus virus inside the genus name of this HIV virus is lentivirus. And when we look this HIV virus through the electron microscope, we can see a spherical shaped structure. So the structure, the shape of this HIV virus is spherical shape. See, this is the structure of HIV virus, spherical shape. Understand of you? Then the size of the HIV virus is 100 to 120 nanometer in diameter. 100 to 120 nanometer in diameter. Understand of you? So the genus name is lentivirus genus name. Then the shape of this HIV virus is spherical shape and the size is 100 to 140 nanometer. 120 nanometer in diameter. Understand of you? Then listen very carefully. See, like all the other virus, in HIV virus also, in the center region nucleus is present. Okay, center side the core nucleus is present. Understand of you? Then the outer surface of this HIV virus is covered with an envelope. Name of this envelope is called lipoprotein envelope. Name of this protein is called lipoprotein envelope. There are two types of glycoproteins are present in this envelope. One glycoprotein name is called glycoprotein 41 and the second glycoprotein is called glycoprotein 120. So there are two types of glycoproteins are present. One is this one, okay, this is one and this is the next one. So one glycoprotein is called glycoprotein 41 and the second one is glycoprotein 120. Understand of you? Understand? Then in the center region, two single stranded RNAs are there. Okay, two single strands. See, this is one single strand, this is the next single strand. So, two single strand RNAs are present in the center region of the virus. Understand? Then, three important enzymes are present in this virus. One enzyme name is called reverse transcriptase. This reverse transcriptase is attached with the RNA. And the second enzyme name is called protease and the third one is called ribonuclease. So, reverse transcriptase, protease and ribonuclease, these are the three enzymes present in this HIV virus. Understand of you? Then one more envelope also there. This envelope name is called matrix protein. One more envelope is there. This envelope name is called matrix protein. And the main function of this reverse transcriptase enzyme is when the cell is entered inside our cell or inside the host cell, this RNA is converted into DNA. The RNA is converted into DNA. So the conversion of this RNA into DNA is the main function of this reverse transcriptase enzyme. Understand all of you? Understand? So hope you all understood all these points. So that's all about the structure of virus. So once again I repeat, the genus name lentivirus, size 100 to 120 nanometer and the shape is spherical shape. Understand of you? Then the next point, yes, the nucleus is present in the center region and outer surface, a envelope is there, lipoprotein envelope. It has two types of glycoprotein, glycoprotein 41 and glycoprotein 2120. Understand? And the nucleus is two single stranded nucleus, RNA. Sorry, the RNA is two single stranded RNA. And it is attached with the enzyme called reverse transcriptase enzyme and two more enzymes also there called protease and ribonuclease. Understand of you? Then one more envelope is there. Name of this envelope is called matrix protein. Name of this envelope is called matrix protein. Understand of you? So now we completed the structure of HIV virus. Now the transmission of HIV virus. How this HIV virus is transferred from one person to another person. See, actually the location of inside the host organism's body, the location of this HIV virus is macrophages. Here macrophages is one of the very important cells of our immune system. So first this HIV virus enter inside the macrophage of the host cell. Here host cell means our body cell is the host cell. So first enter inside the macrophages. Understand? Then inside the cell, this virus can live 1.5 days. Okay, 1.5 days this virus can live inside the host cell. And outside the cell, this virus can live only 6 hours. So inside the cell it can live for 1.5 days and outside the cell it can live for 6 hours. Understand? So now I said the location where it is located. It is located or enter inside the macrophages. Then the lifetime of the HIV virus, I said the inside the cell the lifetime is 1.5 days. Outside the cell only 6 hours. Understand of you? Then the next point is transmission. How this HIV virus is transferred from person to person. See, maximum this HIV virus transfer from one person to another person by many ways. Okay, the most important way is unsafe sexual contact. Okay, unsafe sexual contact is the major reason for the transmission of this HIV virus from one person to another person. Understand? Then sharing the common needles for putting injection and taking some illegal drugs. Sharing the needles. Okay. The third transmission is, say, blood transfusion. Without checking the blood properly, the blood is transferred from one person to another person. When the donor is infected by this virus, the recipient also infected. Okay. So if the blood is not checked properly before the transfusion, it leads to AIDS. It leads to spreading of this disease. Understand a few? So now I said many transmission method. Then say contaminated needles. Here contaminated needles means, see, the needles contaminated with this HIV virus. When a person using this contaminated needle, that person is also affected. Then sometimes this HIV virus also transfer from the mother to fetus. If the pregnant mother is infected with this HIV virus, this HIV virus can enter in the body of the fetus. So these are some of the transmission methods. This. Lesson a few. Lesson. So now we completed the structure and transmission. Listen the next point very carefully. See. Once this HIV virus enter inside the macrophages. Okay, enter inside the macrophages. So just to imagine this is the macrophage. Once this HIV virus enter inside the macrophages, immediately the RNA of this HIV virus is transferred into DNA or converted into DNA with the help of the reverse transcriptase enzyme. Already I told you the nucleus, one enzyme is attached inside the nucleus, name of the enzyme is called reverse transcriptase. So when this HIV virus enter inside the macrophage, the macrophage means our body cell. Okay, so when this HIV virus enter inside this macrophages, immediately the RNA of this HIV virus is converted into DNA. Okay, so just imagine the DNA of the HIV virus. So this DNA is mixed with our cell's DNA. Already inside the macrophage also DNA is there. Okay, so this HIV DNA and our DNA both are mixing. Okay, both are incorporated. Now these two DNAs togetherly producing the other particles of the virus. Other particles means this envelope, then glycoproteins. Okay, so these other particles are produced inside our macrophages. Once I repeat this point, when this HIV virus enter inside our host cell, immediately the RNA is converted into DNA with the help of this reverse transcriptase enzyme. Okay, so now both DNAs are mixed. Now the viral particles also produced inside the macrophages. Understand? Now this virus is entering inside the T helper cells. Just imagine it's the T helper cells, T cells. So now the virus is leaving from the macrophages and it is entering inside the T helper cells. Okay? After reaching the T helper cells, this virus multiply. Okay, and produce many progeny. The virus producing many progeny. So here this macrophage is act as a factory for HIV virus. Okay, factory for HIV virus. So after entering inside the T helper cell, they increasing their number. Okay, the progeny of the N1 virus are produced inside the T helper cell. Finally, these T helper cells are breakdown or destroyed and going to attack the other T helper cells. Like that, all the T helper cells are destroyed inside the body. All the T helper cells are destroyed inside the body. So when the T helper cells number is decreased, automatically the acquired immunity power of the human also decreased. Okay, so when the T helper cells number is decreased, automatically the acquired immunity power also decreased. So during this period, when acquired immunity power is slowly decreasing, that particular person suffer from different types of symptoms. Okay, fever can easily attack that particular person, weight loss, okay, diarrhea, so continuous cough. So these are some of the symptoms of this HIV disease, AIDS disease. Understand of you? Understand? So finally the person is unable to get any protection, any immunity power against any infection. So any infection can easily attack that particular person because acquired immunity power is completely reduced because of the reduction in the number of T helper cells. Understand, my dear students? Understand? Okay. Now we completed the transmission method also. Then how we can identify this AIDS disease? Okay, already in the lower classes we have studied there are two tests are available. The first test is called ELISA test. Okay, so ELISA test, and the second one is called Western blot. Here is ELISA test. So with the help of this ELISA test we can easily identify or detect the HIV antibody. If HIV antibody is produced inside our body, it indicates HIV virus is present inside the body. Okay, when HIV virus is entered inside the body, immediately our immune system producing HIV antibodies. So if any antibodies are there in the blood or in the body, it indicates the person is infected with the HIV virus. So this is the ELISA test. Okay. Second one is a Western blot test, confirmatory test. Isn't it? So this test is used to detect the viral core protein. The proteins of this virus, if the proteins of this virus is present in the person's blood or body, it indicates the HIV virus is there. So ELISA test and Western blot, these two are the important tests used to identify this HIV virus or detect this HIV virus. Understand of you? Then the last topic is prevention. How we can prevent this HIV disease or the AIDS disease? See, the first one is avoid unsafe sexual contact. Okay, so it's a major prevention method. So avoid unsafe sexual contact is the major prevention method. Then, before transferring the blood from one person to another person, the blood should be checked thoroughly. Okay, already I told you, if a person is infected, when we transfer the blood from that infected person to a normal person, the normal person is also getting a chance to getting this infection. Okay, so before transfer the blood should be checked properly and use disposable syringes and needles. Okay, so don't share the common needle. Okay, don't share the common needle. Then many awareness programs nowadays, the government, NGOs, then World Health Organization. So these organizations conducting many awareness programs. So we have to participate in this awareness program also. Then only we can get more information about this AIDS disease. Understand of you? So these are some of the prevention methods. Okay, my dear students. So now we completed one important immunodeficiency diseases. So immunodeficiency diseases meaning I told you failure in one or more components of our immune system leads to some diseases. These diseases are called immunodeficiency diseases. Okay, my dear students. Then our next topic is hypersensitivity. Okay, hypersensitivity. Hypersensitivities. In common we can say allergy. Okay, commonly we can say allergy. Say hypersensitivity meaning. Say sometimes our immune system producing excessive immune response against a common antigen present in the environment. Okay, so hypersensitivity meaning it's a excessive immune response produced by our immune system against a simple or common substance present in the environment. So this condition is called hypersensitivity, otherwise called allergy. So some simple substance present in the environment can cause some severe reaction in the body of some sensitive person. In the body of some sensitive person. So this condition is called hypersensitivity. This condition is called hypersensitivity. So the substance which are causing this hypersensitivity or substance which are causing this allergic reaction is called allergens. Example, many examples we can say the dust, pollen grains, some medicines, venoms of some insects. Okay, so these are the some of the examples for the allergens. Understand? Allergens. Some of the examples for allergens. Then there are many examples are there for allergy. Hay fever, asthma. These are the examples for the hypersensitivity or allergy. Understand you? So the substance which are causing or which are producing this allergic reaction is called allergens. It is called allergens. And example for allergy is, what are two examples I said, hay fever and asthma. Then symptoms. There are many symptoms are there. Difficult in breathing, sneezing, watery eyes. Okay, so these are the symptoms of this allergic reaction. These are the symptoms of this allergic reaction. So I hope you all understood these allergic reactions. Then one very important one more question. See, in the last class we studied about antibodies. There are 5 types of antibodies are there. IgA, IgG, IgM, IgD, like that 5 antibodies are there. Here, inside our body, this allergic reaction is produced by one type of antibodies. Name of that antibody is called IgE antibody. So this IgE antibody will be producing excessive immune response against a common substance of the environment. So IgE antibody, then one more cell, the mast cells. Okay, so the mast cells are also one of the reason for the allergic reaction or hypersensitivity. So this is very important for one more question. Okay, then one more point, anaphylaxis. Anaphylaxis meaning, say, sudden severe hypersensitivity is called anaphylaxis. So in some sensitive person's body, allergy can cause sudden and severe reactions, okay, because of the degranulation of these mast cells. So because of degranulation in mast cells, in some sensitive person's body, this allergic reaction is very severe and suddenly happening. Actually, the duration of this allergic reaction is this allergic reaction is started when this allergens are contact with our body, and it is lasts for maximum 30 minutes. Okay, maximum 30 minutes. Okay, so immediately it is this reaction is takes place after the contact of these allergens. Maximum 30 minutes. So now we completed the second topic, hypersensitivity. So here very important one more question is the reason for the allergic reaction is IgE antibody. IgE antibody. Then the next one is vaccination and immunization. Okay, see, vaccination and immunization. First definition for this word, vaccination and immunization. See, vaccination meaning, see, administration of vaccine to produce immunity power against a particular disease is called vaccination. Administration of vaccine against a particular disease, vaccine to produce immunity power against a particular disease is called vaccination. Then immunization is the process of the body to develop immunity power against the diseases is called immunization. Okay, the process of the body producing immunity power against a particular disease is called immunization. So now we are going to study about this vaccine. Okay, study about vaccine. See, vaccine is a biological preparation. It is prepared from some microorganism. Vaccines are prepared from some microorganism or prepared by using microorganism. And this vaccine giving active acquired immunity power to our body. Okay, in the last class I told you acquired immunity is divided into two types, active acquired immunity and passive acquired immunity. Here this vaccines are giving active acquired immunity to our body. Listen of you see. When a vaccine is injected in our body, immediately this vaccine teach our immune system how to protect our body from the disease. So the main function is teaching our body. The vaccine which is entered inside the body is teaching our immune system to protect the body from the diseases. Understand? In some case, some additional dose of vaccine also should be given. Okay, for some diseases, 2 or 3 times the same vaccine is injected. So this vaccine is called booster vaccine. This vaccine is called a booster vaccine. Understand, my dear students? Then, now we are going to study about the types of vaccine. Is very important 5-mark question. So listen very carefully. Types of vaccine. See, vaccines are classified into 3 major types. The first type is called first generation vaccine. Second one is called second generation vaccine. Third type is called third generation vaccine. So the vaccines are classified into 3 major categories. First generation vaccine, second generation and third generation vaccine. Understand? Then this first generation vaccine is again classified into 3 types. Okay. The first one is live attenuated vaccine. Second one is killed vaccine, and the third one is toxoid vaccine. So the vaccines are classified into 3 major categories: first generation, second generation, third generation. Then the first generation vaccine is again classified into 3: live attenuated, killed and toxoid vaccine. See, the first one, live attenuated vaccine. See, here some very weak microorganisms or aged microorganisms or less toxic microorganisms are used as a vaccine. Okay, a weak, aged or less dangerous or less virulent microorganisms are used as a vaccine. So these vaccines are called live attenuated vaccine. Understand? Example for this live attenuated vaccine is MMR vaccine, mumps, measles and rubella vaccine. Then second type of vaccine is called killed vaccine. Here the microorganisms are killed by using heat or some other method. So by using heat or by using some other method, the microorganisms are killed, then it is used as a vaccine. Example for this killed vaccine is polio vaccine. Example for this killed vaccine is polio vaccine. Understand you? And the third one is toxoid vaccine. Okay, toxoid vaccine means the toxins or the chemicals taken or obtained from the microorganism is used as a vaccine. So that vaccines are called toxoid vaccine. Example DPT. Understand, student? So live attenuated means the live microorganisms are used as a vaccine. Okay, here the which type of microorganism you have to select to prepare this vaccine? Live but very weak or aged, okay, or less virulent microorganism. Second example for this vaccine is MMR vaccine. Second one is killed. Here microorganisms are first killed by heat or some other methods. Then it is used as a vaccine. Example polio vaccine. Third one is toxoid. The chemicals obtained from the microorganism is used as a vaccine. Example DPT. Okay. Then second one is second generation vaccine. So first generation vaccine we completed now. Then second one is second generation vaccines. Here these vaccines contains pure surface antigen of the pathogen or microorganism. Okay, so pure surface antigen of the microorganism is present in the second generation vaccine. Example for the second generation vaccine is hepatitis B vaccine. Hepatitis B vaccine. The third generation vaccine. Third generation means it's the very purest vaccine and it's a high potentiality vaccine, high capacity vaccine. Understand a few? It's a third generation, it's the purest high capacity vaccine is called third generation vaccine. And nowadays some new evolutions also there in the vaccine field. This new vaccine is called DNA vaccine or recombinant vaccine. Okay, it's a revolutionary vaccine. So now we completed the 3 types of vaccines also. It is very important for 5-mark question. So my students, so today we have completed immunodeficiency diseases and one examples with one example AIDS. Okay, then structure of HIV virus transmission, identification of HIV disease, then prevention. These are some of the topics we studied in this immunodeficiency diseases. So first topic, and the second topics we studied about the hypersensitivity, otherwise called allergy. Okay, there I told you one very important one more question is there. The antibody, the hypersensitivity reaction is created by IgE antibody. Then the third topic, finally we studied about this vaccines, types of vaccine. So this type of vaccine is very important for 5-mark question, and the structure of HIV virus diagram is very important for 2-mark question. Understand, my dear students? So hope you all understood today's class. Today's class is very easy class and very simple class. So try to complete today itself. Okay, we will meet in the next class. Thank you.